So guys, I'm going to go through this pretty nice and quickly. This shouldn't be too bad. The process in which you have a lion and tiger mating together, so two totally different organisms, that's called hybridization. If you have two animals that have a specific characteristic that you're wanting and you make them mate, that's called inbreeding. What causes the DNA to break apart in the first place is restriction enzymes. So remember, in the lab that we did when we made our own gels, the first thing that you did was you took your DNA and you actually added restriction enzymes, and then we left it in the water bath for a while we ran our gel, meaning we set our gel. So we use restriction enzymes to chop up the DNA, so just keep in mind what we actually did in the lab as well. Then what we did in the lab is once our DNA was chopped up, we took the little pipettes and we removed the sections, the samples of DNA, and you guys really carefully took the pipette tips and put them into each of the wells and put the DNA into each of the individual wells in order for this process, gel electrophoresis, to happen. So the gel had those wells, and then you hooked up, we hooked up a power supply. And so if you remember, and I'll just cut to the chase here, this is kind of what it looked like. You had the negative end, which we also call the anode here, and you had the positive end here. And so what that does is it forces the DNA to be pushed through the gel. Okay, so that's what we did, and it separates it based on size, which you'll see in a second. In order to produce more insulin gene or anything more of a specific gene that we're interested in, what we can do is we insert it into plasmids. Now, plasmids are in bacteria until we want those bacteria to grow. So once we've inserted the DNA into the plasmids, because we have two different organisms, DNA together, we call it recombinant DNA. And the way to get rid of the bacteria that doesn't have the plasmid with the recombinant DNA, we put it into antibiotics. And the reason why is that the plasmids themselves actually make the bacteria antibiotic resistant. So the antibiotics don't kill the bacteria that have the plasmids, but it does kill the ones that don't have the plasmids. When we're talking about cloning, what you're doing is you're taking a somatic cell, which, by the way, is diploid. So a somatic cell is any cell that is not either an egg cell or a sperm cell. And you take an egg, remove its nucleus, and insert the nucleus of the somatic cell into the egg cell. Okay, so the egg cell was originally haploid. You took the nucleus of the somatic cell, which is diploid, and inserted it in. And so basically by doing that, you are avoiding the process of fertilization. So you don't need two haploid cells coming together to make a diploid because you're already inserting a diploid nucleus. Okay, here my restriction enzymes chopping up my DNA. Notice this is the longest one, so this is going to travel the least distance, while this is the shortest one, so it's going to travel the farthest distance. You just do it in a row, guys. Don't go crazy drawing them everywhere, okay? And then it says label the positive negative ends. We did that. Remember, this is my anode, this is my cathode, and this is my well, which I injected the DNA into, and then it separated out based on size. Okay? I'm going to go now to the rest of the review, and I'm just going to do this verbally because I don't want you guys to be copying down exactly what I write. Okay, so we're on number 8. What happens in PCR, the first thing we need to do is heat it up, and so you have double-stranded DNA, and by heating it up, they separate. By then cooling it, what that allows to happen is the primers are able to attach to the DNA. And then once it's heated up again, then the DNA polymerase can go in and make another copy of DNA. Primary producers, those are your plants, also called autotrophs, because what they're doing is they're taking unusable energy, so either the sun in the case of photosynthesis or inorganic molecules in the case of chemosynthesis, and they're making it into carbohydrates that we as consumers can eat. Primary consumers, they are herbivores. They only eat plants. Secondary consumers are going to be carnivores because they're eating primary consumers. So an herbivore only eats plants, omnivore eats meat and plants, and carnivore only eats meat. With each organism that is consumed, what happens to the amount of energy that is available? So with each organism that's eaten, you lose energy. And so if we wanted to be as efficient as possible, meaning getting the most energy out of our food as we can, we would just eat plants because The only step in which energy has been lost is photosynthesis. No one has consumed those plants until we do. In the water cycle, if water is going from the oceans to the air, that's evaporation. If it's going from the air back into the land, that's because of precipitation. With the carbon cycle, there's 3 different ways that carbon is released into the atmosphere. So first of all, when we breathe out, our respiration makes CO2, so that's one way. Another way is any kind of volcanic activity or any kind of earthquakes or anything like that where CO2 is released from the ground. And the last one is when we burn fossil fuels. So whenever we drive our cars, anything like that, that is releasing CO2 into the air. And the issue with CO2 is that it causes global warming. Okay. 2 ways that carbon is removed from the... so that's another part of that same question. How is it removed? Well, plants can consume it. For photosynthesis to happen, they require CO2. So the more plants we have, the less CO2 there is in the air, which is awesome. The other way, it can get absorbed, taken into the land. It can be absorbed and dissolved into the water. The unfortunate thing in those cases, when it's dissolved into water or into the land, it can always be expelled. It can always be put back into the air. Explain the 3 different types of symbiotic relationships that you have. So we've got commensalism, in which one organism benefits, but the other one, it has no effect on it. We've got parasitism, in which one is helped and then the other one is hurt. And then we've got mutualism, in which both parties are helped by the relationship. If there were no limits to species growth, they would grow exponentially. So remember I drew on the graph in which you've got population size on the y-axis and over time on the x-axis, and it just goes whoop all the way up. Okay, that's exponential growth. Now, this doesn't happen because every species has a carrying capacity, and so that is the maximum population that can survive. So my example that I gave in class was my classroom has a carrying capacity of 17 kids, because if I have more than 17 biology students at one time, if I have 18 or 19, they will not survive. Okay, and so as a result, I can maximum have 17 kids and them still survive. All right, Lamarck's contribution, he said that traits acquired during an organism's life will then be passed on to its offspring. So the example, the giraffe, it stretches its neck, the extra stretched neck is then passed on to its offspring, etc., which we found not to be the case. Blank is a characteristic that makes an organism better equipped. So this is oh my goodness, I'm doing this blank. Hold on one sec. Sorry, I'll go on to this one because this one was in my head anyways. This is fitness, so it measures someone's ability to survive and produce offspring. For number 2, I don't want to kill time and make this video too long. Guys, look in your notes. I'm really sorry. It's the one right next to fitness. I just don't have my notes in front of me. I'm just doing this at the top of my head. Okay. 4, homologous structures. So homologous structures are structures that we have in common with other organisms. So for example, the bone structure, the fact that we have the exact same order of bones, that they're very similar. So that demonstrates that evolution in fact happened. 5, vestigial structures. Vestigial structures are structures that we have right now, but that serve no purpose for us currently. So they must have in the past, but as we evolved, we lost need for them. So the example I used in class was the appendix. How did the movement of continents affect the diversity of species? So the example I gave was elephants, the fact that the Asian elephant is very different than the African elephant, and it's just based on their environment. So they evolved because the ones that were able to adapt to their environment were the ones that survived. And since Africa and Asia are so different from each other, that's what caused the differences in the organisms themselves. Convergent evolution. This is when you see a similarity and you assume that they're closely related, but it's not the case. So the example I gave was bats and birds. They both have wings, but those 2 traits evolved separately. So bats having wings and birds having wings, it seems like they would be closely related, but they're not. And so that's an example of convergent evolution. Just because they acquired these traits separately and individually does not mean that they are more closely related. 8, did our Earth always have oxygen? No. In fact, originally it had no oxygen, and oxygen was actually toxic for a lot of animals. And then when you get these photosynthetic organisms that are making oxygen, it caused the death, the destruction of a lot of populations of organisms because they couldn't survive with oxygen. That did allow us, however, to evolve. So good for us, bad for them. In the endosymbiotic theory, what's happening, originally our Earth was just populated by prokaryotes. And 2 of those prokaryotes were mitochondria and chloroplasts. And what happened was that one prokaryote goes and it engulfs, it eats a chloroplast, or it eats a mitochondria. And all of a sudden, rather than, causing it to break down and actually eating it, what happens is a relationship, is a mutualistic relationship in which the mitochondria is benefited from the protection in the prokaryotic cell, and the prokaryotic cell gets energy from the mitochondria or the chloroplast. And so that's how you get the origins of eukaryotes in the first place, the fact that one prokaryote actually consumed another one. Okay? So going back here, guys, please check for this one. My head's not working right now, but otherwise I've given you all the solutions for this, and good luck on your final.